This is chapter 3, which is going to focus on microscopy. This cover slide is, as you can probably tell from the gentleman's funky pants there, this is kind of an old, I guess, drawing of an individual in front of a very large piece of equipment that is an electron microscope. And I chose this picture because when I was going to UC Riverside for my PhD, and I studied a virus for my PhD, and so I used electron microscopes to view my virus, and I utilized an electron microscope that looked pretty much just like this. And so they are quite large, so that's why I liked this diagram that just kind of showed that. Although, again, this is older. They have gotten smaller over the years. Like they do make a tabletop electron microscope now that does fit on a table, but we're still talking about a fairly large table that it takes up. And they're incredibly expensive. The tabletop electron microscope costs about $300,000. So before we get into the different types of microscopes, it's important to note the different sizes of organisms and kind of know what the capabilities of the different types of microscopes are. So this diagram is showing you here a tick, kind of starting here on the left with the larger size and then going down to small. And then you can see on the bottom you have kind of a size scale here along the bottom from meters down to picometers. And then you also have here indication of different ways to view. So here's this first kind of bracket here is what we can see with just our eyes without a microscope or anything. So we can see a tick, but it is quite small. It's towards, the point where we don't have that great of vision that small, but we can definitely see it. But then you get to red blood cells. They're obviously not able to be seen with the naked eye. We need a light microscope. That's what the LM stands for here. And then E. coli bacteria. Now we can see E. coli with a light microscope, and that's what you see here, that it's still following in that range. However, the picture they're showing us here, they've used a scanning electron microscope, or SEM, which will give you a much more magnified, much higher quality picture of the bacteria. Here are viruses called bacteriophages. They're viruses that infect bacteria. And viruses are in the nanometer size range, and so they can only be viewed with an electron microscope or other high-powered microscopes. You cannot see viruses with a light microscope, as you can again see from this diagram what it's depicting. And then on the far end over here, you have DNA, which is quite small in the nanometer to picometer size range. And even you can see it with some electron microscopes, with the transmission electron microscope, but your better view often is with what's called an atomic force microscope, or AFM. So this is showing the, again, just kind of giving you an idea, hopefully this is something you learned before, of just the different measurements that are utilized. Of course, we use the metric system in science. So we're talking about meters, millimeters, micrometers, nanometers, picometers. Okay, so you do want to have a basic understanding of the relationship of these measurements. If you watched the video, one of the videos that I had in chapter 1, then you saw this microscope already when they talked about Van Leeuwenhoek and how he made his own microscope, made a very simple microscope, and was able to see a lot of things that no one had ever seen before, including bacteria. So even though it's a very simple microscope, it was effective and was did allow for viewing bacteria. So light microscopy is any type of microscopy that uses visible light. And so these are all the different types of light microscopy, and we will go through each one, and you will see if you look at the study guide that there is kind of a focus for you, certain things that you want to be able to know about each type of microscopy, the general characteristics, kind of the pros and cons. What is the microscope good for? What is it not good for? That's kind of going to be the focus for the different types of microscopes. So the first one is the compound light microscope. This is the type of microscope that we use in the microbiology lab. You could see all, the parts labeled here. This is something, again, if you look at the study guide, you're going to see that you need to be able to label a microscope. This is something you would get tested on kind of double. You could get tested on this for lecture in this chapter, but also in lab from your microscopy chapter. So this is an animation. Let's see if it'll play. I don't know if I have my volume on. Oh, let me see real quick if I can get that. Okay, see if we can get back to full screen. Here we go.
Light microscopes are useful for studying specimens ranging in size from about 1 millimeter to about 200 nanometers. It's impossible to study anything smaller by light microscopy because the shortest wavelength of visible light is about 390 nanometers, and anything smaller will not interact with the light. The most common form of light microscopy is called bright field microscopy. In this form of light microscopy, the light beam passes through the sample and into the objective lens. Let's look at the parts of a light microscope to see how a specimen is magnified and viewed. The lamp is where the light beam is initiated. This beam is divergent, spreading out as it approaches the condenser lens. The condenser lens gathers the beam of light and focuses it so that it converges on the specimen with high intensity in a small area. The specimen usually is on a glass slide that rests on a platform called the stage. When the light beam hits the sample, the light rays are either reflected or refracted or absorbed by the sample. The beam then continues to diverge upward on its path away from the specimen. After the beam leaves the specimen, it passes through the objective lens. The objective lens performs the bulk of the magnification of the specimen. The eyepiece receives the light beam from the objective lens and refocuses it to recreate the image in the viewer's eye. Lenses found within the eyepiece are called ocular lenses.
Okay, so that was a very nice summary and kind of showed you an important concept that this diagram also shows you, which is the path of light through a compound light microscope. Another important concept is to know that total magnification is calculated by multiplying whatever the magnification of the objective lens that's being used by the magnification of the ocular lens. Generally, ocular lenses are 10x. That's pretty standard. But your objective lenses, it does depend on the microscope, but usually they have 4 lenses, starting with the lowest being a 4x, then a 10x, then a 40x, and then a 100x. So you want to be able to calculate a total magnification if I were to tell you which objective were being used and that the ocular was a 10x. So let's say if you were using the 40x objective and the ocular is 10x, then the total magnification should be 400x. Important concept with microscopy is resolution, which is the ability to resolve or to distinguish especially to anything that's really close to each other. So when it says 2 points, it's like if there's 2 points in a specimen that are very, very close to each other, if you have good resolution, they will be able to be distinguished. But if you have poor resolution, you're not going to be able to distinguish those as 2 separate entities, and it's just going to look like a blurry blob. And the animation did mention the resolution capabilities of a compound light microscope, and why, because of the resolution limitations, why the microscope can only view up to a certain size, and why then it does not work to visualize viruses, which are smaller than that. As far as light microscopy goes, shorter wavelengths of light provide greater resolution. So we'll compare light microscope resolution to electron microscope resolution. There's quite a difference because electrons have much shorter wavelengths than does light. And so you get much greater resolution with an electron microscope, which is why electron microscopes can visualize really small things like viruses and DNA and proteins and things like that. So this is a nice kind of, I guess, analogy, if you will, of that wavelength. So picture A here is showing visible light, and these large yellow circles are representing the longer wavelength of visible light, and how it's just not that penetrating. It cannot penetrate between the gaps between the fingers here to resolve, and so that's why the hand appears as it does. You can't resolve the fingers. But electrons with much shorter wavelengths are very penetrating and therefore provide much greater resolution. Refractive index is another important concept when it comes to light microscopy. It has to do with the fact that light bends when it hits air, and because of that, you can lose resolution because the light, once it passes through the specimen, it refracts or bends, and it doesn't enter, all of it does not enter the objective lens, and therefore that leads to loss of resolution. So the reason that we use immersion oil when we use our 100x oil lens, our objective lens, is that oil keeps the light from refracting and therefore maintains that resolution or increases that resolution that otherwise would be lost. And you can see that concept here. So without oil, you could see how the light would refract and not enter into the objective lens, and therefore resolution would be lost. But with oil, the light is able to continue the path from the specimen into the objective lens, and therefore it can increase the resolution. So here's a question that I will have to figure out how to convert over to the built-in questions, since we don't get to do open-ended questions on those quiz questions built into the video. But this is an important concept that you do want to be able to address as far as the study guide and the exam goes. So you want to be able to indicate the path of light from the light source all the way to your eye through a compound light microscope. So as the animation mentioned, bright field is the most common type of or common use of compound light microscopes. It's generally what we use in the microbiology lab. That's where you have a bright background and the specimen has to have contrast. It has to appear darker against the bright background. So often, because most microbes have no color to them and no contrast to them, we have to stain the microbes to give them that contrast. So we will also cover staining in this chapter. On the contrary, though, you do have dark field microscopy, which is still using a compound light microscope, but now what happens is you put a filter over the light source and it creates a dark field, a dark background. And this is useful for viewing unstained specimens because they will appear lighter against a dark background. And then you have phase contrast microscopy, which is very similar to compound light microscopy. The only difference really is that you have these extra lenses. They're, I don't know if I should say lenses, filters, I guess I should say is the more correct term. That these filters can be put into place, and usually they're got numbers on them. It's phase 1, phase 2, phase 3, etc. And you can choose which filter is best for the specimen being viewed. But you can see from the image here, and all three of these images so far have been the same organism, a paramecium, which is a single-celled eukaryote in the protozoa group. And so you could kind of just compare the different, appearance that you can get with the different type of microscopy. So this is unstained, but notice that it's, of the three so far, I think it's probably the most detailed, especially when you see all the cilia here on the outside, and some of the internal structures. So what happens with the different filters of the phase contrast is it kind of can even add some dimension to it. It can add some color to it, even though it's not actually a stained specimen. Here's differential interference contrast. Still looking at a paramecium. Now you could really see that this is the best image thus far, right? This is another way where it's similar to phase contrast, where it can give more contrast, more dimension, and more color to the specimen without actually having to stain the specimen. The specimen can be viewed still living and unstained, but it really can give a much more detailed image, and that's because it uses two beams of light and prisms to have different light beams and different colors of light kind of hitting that specimen and enhancing the image that results. Now here's the first picture where we'll see a different specimen being utilized rather than the paramecium. We'll come back to the paramecium in a minute, but they're using something different here. They're using an organism that is a spirochete, one of those spiral-shaped bacteria. It's called Treponema pallidum, and it's the cause of syphilis. And they're using this because the bacteria is very, very thin, and it's really hard to see under a regular light microscopy, whether you use bright field, dark field, etc. It's very thin and hard to see. So what they do here is they use a fluorescent dye to stain the bacteria. And then the microscope, it is very much like a compound light microscope, but instead of regular light as the light source, it's an ultraviolet light. And when UV light hits the fluorescent dye, it excites the molecules and they fluoresce. And thus you would see these spiral-shaped bacteria fluorescing under the microscope. For confocal microscopy, you're again seeing fluorescent dyes being used. This also usually will be used in conjunction with a computer to create a three-dimensional image. We're going back here now to the paramecium again, but you can see quite a difference here, with the fluorescent colors, with how detailed the cilia are. So this is, much more powerful of a type of light microscopy. It's using different wavelengths of light, and again, in conjunction with the computer and with fluorescent dyes. And here's another one, even getting more powerful. Again, using fluorescent dyes, using different wavelengths of light. And again, it doesn't mention it, but I think most likely in conjunction with a computer. So those were all the types of light microscopy. This is not a type of light microscopy because, as you can see in the name, scanning acoustic microscopy uses sound, right? Acoustic. So this measures sound waves that are I don't know what you call it, directed at a specimen, I guess. And then when they bounce off or reflected off of the object, it can actually create a map of the specimen, an image. And so this is useful for studying biofilms, for studying cells that are attached to a surface. And that's what we're actually seeing here in this picture. So this is not paramecium. This is looking at a biofilm. And then, oh, that one might have been out of order because now we're going back to a light microscopy. Super resolution light microscopy. I don't think I have this one on the study guide. This one is only in our newest edition of the book, the 13th edition. So if students are using an older edition of the book, they wouldn't see this in their chapter. And so I did leave it off of the study guide for that reason. So moving on to electron microscopy. So in electron microscopy, like I told you, it uses electrons instead of light. Electrons have much shorter wavelengths than visible light, and so you get much greater resolution. So this is great for viewing viruses that are too small to be visualized with a light microscope. You can see that the magnification abilities are much greater here, from 10,000 to 10 million times resolution. I can't see this little box. It's in my way. I think it says 10 picometers for the resolution. So there's two types of electron microscopy. You have transmission, and that's, I think, actually doesn't indicate, but I think that might be referring to transmission because it is more powerful than is the scanning electron microscopy. So you have transmission electron microscopy, which is more of, gives a better internal view, and then scanning, as the name kind of implies, it more scans the surface and is better at giving more of the external surface view. So we're going back to, again, looking at the paramecium specimen so you can see how much more powerful these microscopes are. You can also see that the structure of these microscopes is quite different than a compound light microscope. And then here's the scanning electron microscope, not as powerful as the transmission electron microscope, not as high of a resolution, but still plenty good to view viruses. So either type of electron microscope is able to visualize viruses, but the transmission electron microscope is much more powerful and can also be used to visualize things like proteins and DNA. So you can see here in the diagram how the scanning electron microscope is better at surface, and now you're seeing all these cilia on the surface of that paramecium. So here's an animation.
Electron microscopes use electrons to probe a sample. Electron microscopes are useful for studying samples in the size range of 10 nanometers to 100 micrometers. This includes specimens as small as ribosomes, up to specimens as large as red blood cells. Transmission electron microscopes, such as the one shown here, are used to produce two-dimensional images of internal structures of cells, while scanning electron microscopes are used to produce three-dimensional images of the surface of specimens. Electron-dense compounds, such as heavy metal salts, can be used to stain specimens for electron microscopy, increasing the contrast and resolution. Let's look at the parts of a transmission electron microscope to see how a specimen is magnified and viewed. The electron gun initiates a beam of electrons. This beam is divergent, spreading out as it approaches the condenser lens. The condenser lens gathers the beam of electrons and focuses it so that it converges on the specimen with high intensity in a small area. The condenser lens in an electron microscope is electromagnetic. The electron beam next passes through the specimen. In a transmission electron microscope, the specimen is a tiny slice of cells that rests on an equally tiny copper grid. This grid is inserted into the microscope at the location shown here. When the electrons interact with the sample, they are either reflected and refracted or absorbed by the sample. The beam then continues to diverge downward on its path away from the specimen. After the beam leaves the specimen, it passes through the objective lens. The objective lens performs the bulk of the magnification of the specimen. The projector lens receives the electron beam from the objective lens and refocuses it to recreate the image that the viewer sees in the fluorescent screen. An internal camera is used to photograph the specimen. You may have noticed that these two types of microscopes share similar parts. Both have a source of light or electrons, a condenser lens to focus this source, and an objective lens to refocus the image. However, you may also have noticed that the path of light is from bottom to top in the light microscope, while the path of electrons is from top to bottom in the electron microscope. All types of microscopy are concerned with resolution. The resolution of an image is the ability to distinguish between objects that are close together. Resolution depends on the wavelength of the source, with smaller wavelengths leading to higher resolution. The average wavelength of white light is 550 nanometers, while the wavelength of electrons in the electron microscope is 0.01 nanometers. This is why electron microscopes are able to resolve objects much smaller than light microscopes.
Okay. So moving on, we have just a couple more types of microscopes to talk about in this recording. You have scanning tunneling microscopy, and these are even more powerful than the electron microscopes. These can resolve down to the atomic level. So I believe this is DNA that we're seeing here. And then the atomic force microscopy, I mean, it's in the name, again, can resolve at near atomic detail. And here, this is protein that we're seeing in this specimen. So here's a question that you will see right now in the recording. Which microscope uses an ultraviolet light source? So staining is the next topic. I'm going to stop this recording and restart a recording as a part 2 for starting with this slide here and this topic.